Okay, let's get started with figuring out how to find knowledge gaps so we can start to write our proposals. And just so we're all on the same page, what I mean when I say knowledge gap, it's some kind of research topic or question that hasn't been investigated or hasn't been answered yet. So if we think about this puzzle piece analogy, all of these existing pieces are previous work. So basically what's already been done. And then we have this one missing piece here, and that's what that knowledge gap is. So that's our uninvestigated, unanswered question. And so that's what we're trying to figure out is what can we study within the context of what's already been done that can fill in these missing pieces, essentially. And that's important because only novel research ideas that address knowledge gaps are the ones that are funded. So if you're writing a proposal and you're proposing something that's already been done, it's not worth spending money on or really even doing that experiment because we already know the answer to it. That's the whole goal of science, is to advance knowledge and to do experiments to give us new answers and new data that we didn't have before. So one example of how you might see phrasing related to a knowledge gap that's being investigated in a research proposal. This is from a proposal I wrote, a project that one of my master's students is working on right now. So what I have in red here, this is describing what's already known, essentially. So what previous work has been done. So this is providing the context. So it's always important to acknowledge what has been done. And so in this particular case, what I wrote is that previous research has demonstrated a relationship between the resident microbial community, so bacteria and archaea, and host thermal tolerance. So this is a microbiome study, just like what we talked about in the first DNA subway lab. So it's important to first acknowledge what has been done. And then in blue here is basically where you have that knowledge gap. So what is unanswered? And what this statement says is, so we've already said previous research has shown this, but the microbial contribution to heat stress response has not been studied in red abalone. So we're basically saying we know from previous studies there could be a relationship here, but specifically that has not been investigated in this one particular organism, red abalone. And in this case, it's important to investigate it in this particular organism because red abalone are actually economically, commercially important. So there's a recreational fishery for them, or there usually is, but they've actually closed the fishery because populations in the wild aren't doing well. So this is just one example of how when you're writing your proposal, you can phrase things to make it clear that what you're studying hasn't been done yet and it's important and we need to do it. And like I mentioned in the overview module, I know that identifying knowledge gaps is hard. That was for me one of the hardest parts of my PhD, was figuring out what I was going to study. And it took me a couple false starts to actually get there. So, I mean, I know when you're reading papers, it can be really easy to be overwhelmed with all these papers, right? Just papers and papers, and you don't really know where to start. And so what I'm going to do in this module is give you a couple possible strategies for how to identify knowledge gaps. And these strategies are going to differ in your starting point. So you have to start from somewhere, right? And so what I'm going to suggest is that you use several different starting points. On the left, I'll go into each of these in more detail in the rest of this video, but you can start with a review paper. You can start with a topic of interest, or you can start with an urgent issue. So this is red abalone, like I just talked about in the research in my lab. We're studying red abalone in part because it's this urgent economic issue and they're not doing well right now. So those are three different possible strategies, and we'll start with this one right here and then we'll walk through the rest. So the first strategy I'm proposing is to read molecular ecology review papers. So first of all, a review paper is unique in that it normally in a traditional quote-unquote research paper, you're presenting some new data and new findings, right? In a review paper, what you're doing is summarizing existing studies. So you're not presenting new data per se, but review papers are a great way to figure out what has already been done in the field. And the really nice thing about review papers is they often have a future direction section. And this is like exactly what you're looking for, because what it will tell you in these future direction sections is in this given field, what still needs to be done. And basically, a future direction is a knowledge gap. The authors of the review paper are saying these are important questions we still have that haven't been answered yet. So that's exactly the type of thing that you want to look for when you're reading a review paper, are these future directions. And I will provide some links to some potential starting review papers in the module this week. And just to note, this strategy is actually how I figured out my dissertation topic. So I was reading a review paper about local adaptation in marine invertebrates, and there was a table that summarized all the previous studies that had been done. One of them was on the snail that I currently work with, and basically that was the starting point for me to figure out that there was this unanswered question in terms of how do different populations of this species respond to temperature stress that hadn't been looked at yet. So that's what I continued to study for my PhD dissertation. So I can tell you firsthand that this strategy does work. A second strategy would be to build on previous research for a topic or study organism of interest. And so for this, I would encourage you to go back to that discussion assignment you did in the beginning of the semester where I asked you to think about potential topics for your GRFP proposal. You already have thought about things you're interested in, either topics or organisms, and you can start there. So what you can do is if you have a particular, for instance, study organism of interest, say you're really interested in tigers, what you can do is do a search for papers that have been written about tigers. So you could do Google, how do we spell that? Scholar, or Web of Science. Those are both good places to look for scientific papers. You could use search terms like tiger genomics or tiger molecular ecology, something like that. And you can look for papers that have already been written about what you're interested in. And then from there, so this is one example of a paper that came out a couple years ago where they looked at the genomics of tigers, and they actually used whole genome sequencing, which we've talked about previously. And once you find one of these papers, you can skim it, and just similar to like we talked about with the review papers, what you're looking for are sections or statements in the discussion or conclusion that explicitly state for you either limitations of the current study and or things that they need to do next based on what their current findings are. So this is an excerpt from this actual paper in the discussion, and it says the origin of the South China tiger remains unresolved since only one specimen from captivity was used here. The validity of this scenario can only be tested with a larger data set, particularly one including wild origin museum specimens in the future. So this right here, again, is kind of one of your aha moments. This is telling you explicitly what a knowledge gap and worthwhile future direction is for this particular type of work. Another potential strategy related to strategy 2 in terms of building on previous research is to go back to what I introduced in one of the previous modules where I had this example with 2 different papers on the same study organism. Yes, that same snail again. If you're thinking I'm going to run out of snail pictures, don't worry, I have a lot. So in this example, we had this study from 2010 that was done using a single gene marker, single gene sequencing, and then in 2016, I used similar populations of this same species, but I used high throughput sequencing, specifically RADseq. So that's another way to build on previous research, is to use new techniques or new types of molecular markers to update studies that have been done previously. So even if people have investigated a question that you're interested in, that doesn't necessarily mean you can't still do that study. If they've looked at that question in the past, for instance, using single gene markers, it's possible for you then to propose to take a more holistic approach and use high throughput sequencing or whole genome sequencing, something like that. Because as we've talked about before, high throughput sequencing really gives you a more complete picture of what's happening because you're using so much more genetic data, so many more genetic markers throughout that organism's genome. And then lastly, for a third potential strategy is to focus on urgent hot topics or species. And the reason why this is a potentially useful strategy is that these types of studies are more likely to be funded because you're proposing to look at endangered species, for instance. So all 3 of these species shown here on this slide are endangered California species. So this is a fairy shrimp, kangaroo rat from his legs there. Dr. Collins in our department actually studies kangaroo rats if you're really interested. And this cute little guy is a tiger salamander. So if maybe you don't want to start with a review paper, you don't really know what topics or organisms you're interested in right off the bat, you can try and figure out if there's an endangered species, especially for California, that might be interesting to you, and then you could pursue research in that avenue. And I would approach this the same way I talked about for the last strategy. Once you have an organism of interest, you could look for papers related to that organism and then try to look in the discussion sections for knowledge gaps. And I'll put a link to a page I found that lists all of California's endangered and threatened species. I'll put a link to that as a resource for this module. So hopefully that gives you some ideas for where to start, and then continue on in this module. That one assignment that I mentioned will be for you to actually put this into practice and to use one of these strategies to find a potential knowledge gap that you're interested in that you could then write your proposal about.